Neurons. They allow our central nervous system to receive messages from the environment and respond as needed. Hi, and welcome to our presentation on the nervous system and homeostasis. Today we'll be focusing on nerves. So living cells can only survive and function within a very narrow range of conditions, and our body maintaining this internal stability is known as homeostasis. And our body's internal environment needs to stay stable despite variations in the external environment. So human survival depends on a system of communication between the internal environment and the external environment. And this is maintained by the control of the nervous system and the endocrine system. So they respond both to internal and external environmental variations. And this is made possible by electrochemical messages relayed from the brain, controlling our nervous system, or by chemical messengers, our hormones traveling through our blood. So the main function of the nervous system is to receive information and carry it to our central nervous system. Interpret impulses and make decisions about responses, to sort the impulses and set priority for actions. So, do we walk across the street, meet a bug, get hit by a car? Like, what's going to happen? And then transmit impulses to the motor units, our muscles, which carry out any appropriate actions. When we look at our nerves, there are two main types of nerve cells found in the nervous system. We have glial cells, which are neurological cells that are non-conducting and used for structural and nutritional support. And then we have neurons, which are neurological cells that carry and conduct impulses. Neurons are the basic unit of the nervous system. A nerve is actually a bundle of individual neurons, and neurons are composed of a cellular body and a number of branch-like extensions. These branch-like extensions are known as dendrites on one end and axons on the other. So dendrites are short thread-like branches that surround the cell body of a nucleus, and they carry impulses towards the cell body. So they pick up information and take it towards the cell body. Axons are longer branches that take impulses from the cell body and carry them away. And typically they're enclosed in a fatty sheath known as a myelin sheath. And a myelin sheath is made up of Schwann cells with small gaps in between. And those small gaps are known as the nodes of Ranvier. So the myelin sheath actually increases the speed of nerve transmission because the Schwann cells insulate the nerve and allow for transmission to occur only at the nodes of Ranvier, which prevents the loss of any charged ions and allows for faster neural transmission. And this also inhibits ion movement in the axonal membrane. So it is a very effective way at increasing neural transmission. So here's an image of a neuron. On the right-hand side, we have our nucleus with our purple dendrites surrounding it. And that, again, carries impulses towards the cell body. And then the axon is a black line surrounded by red myelin sheath made up of Schwann cells. And in between those red Schwann cells, we see the small nodes of Ranvier. And this is the place where neural transmission or neural conduction will occur. And then the axon ends in axon terminals, which will then send a chemical impulse on to the next neuron to their dendrites. So myelinated neurons form the white matter in the brain and spinal cord, while the cell bodies and unmyelinated neurons form the gray matter. Myelinated nerve fibers are covered with a neurolema, which, along with the Schwann cells, promote the regeneration of the nerve fiber after trauma. So they help our cells regenerate. Unmyelinated nerves lack a myelin sheath, and these are not able to be regenerated after any injury. So a condition of the nervous system is multiple sclerosis. And this causes hardened tissue to form on top of the myelin, which affects the nerve transmission. So nerves aren't able to transmit as easily. The more scar tissue there is, the worse the symptoms of multiple sclerosis are. So when we look at neurons, they're classified into four general types. We have anoxic neurons, which are found in the central nervous system. There are no axons, and they have an unknown function. We have unipolar neurons, which have a cell body off to one side. The axons and dendrites are continuous, and they are associated with receptors sensing the environment. So these are typically sensory neurons. We have bipolar neurons, which have a single dendrite and axon with a cell body. Body of a cell is found in the middle, and this is very rare. These are found in the rods and cones of the eye. So bipolar neurons have the cell body in the middle. And then multipolar neurons are several short dendrites with a single long axon with one or more branches. So typically, this is what we'll see in our diagrams. It is most common, it activates glands and muscles, and it is a motor neuron. So here we see all the 4 different types of neurons, the unipolar, the bipolar, the multipolar, and the pseudopolar, which we called anoxic neurons. So here we can see the difference between motor and sensory neurons, where our motor neuron, as we can see, looks like the typical diagram, where the cell body is at one end near the dendrites and the axon terminals are at the other. The sensory neuron has a single line from the dendrites to the axon terminals, and the nucleus kind of hangs off the center. So when we're looking at the conduction of a nerve impulse, also known as an action potential, it has both a chemical and electrical component. So this is an electrical event because the resting nerve is said to be polarized. So it's typically about negative 74 millivolts. There's a relatively negative charge inside the neuron and a relative positive charge outside the neuron. So the difference in this charge is called the resting potential or membrane potential. And this charge difference is caused by the concentration of sodium and potassium ions. There's going to be 3 main components to a nerve impulse. So the first one is polarization. So in a resting membrane, the neuron is positive on the outside relative to the inside, or negative on the inside relative to the outside. So that resting membrane is said to be polarized. And like I said, it has a potential difference of about negative 70 millivolts. A polarized membrane has more sodium on the outside of the axon than potassium on the inside. So when we look at this resting membrane potential, we see that phospholipid bilayer, and we have more positive ions outside compared to the intracellular fluid inside our axon. So the second stage of a nerve impulse is known as depolarization. So when depolarization occurs, sodium rushes into the axon, which would be down their concentration gradient. So this would be passive transport, which now will cause there to all of a sudden be a positive charge on the inside of the axon relative to the outside. So we've depolarized. This stimulation of the neuron by electrical discharge causes those potassium gates to open. So potassium gates are going to open. Sodium gates are going to open, and sodium is going to rush right into that cellular membrane. So this reverses the membrane potential from about negative 70 millivolts inside to about positive 40 millivolts with respect to the outside of the cell membrane. So now our membrane is depolarized. So now once the sodium has rushed in to its equilibrium, the membrane is now going to become impermeable to them as those sodium gates close. The next step of a nerve impulse is repolarization. So once the action potential has peaked, the sodium gates close and now potassium gates are going to open. So now because we have a net positive inside the cell membrane, potassium is going to want to rush out of the cell to neutralize itself again because we now have a relative positive inside the cell membrane. Once that happens, that sodium potassium pump is going to kick into action, maintaining the resting membrane potential of larger amounts of sodium outside of the cell compared to potassium inside of the cell. Now, this entire process only takes about 2 milliseconds. So when we look at this diagram of a nerve impulse, this happens within milliseconds. So about, like I said, about 2 milliseconds. So at the beginning, highlighted by yellow, we see the beginning of depolarization. Now this is when the sodium channels open and sodium will rush into the cell. This still occurs during the green section of this graph. Then, once the impulse has peaked, sodium channels are going to close, potassium channels are going to open. And during that blue highlighted section, potassium is going to rush out of the cell, repolarizing the cell. Then potassium channels are going to close, and a sodium potassium pump is going to maintain that resting potential. Okay. Here's another diagram where we see how that depolarization moves down the axon from the cell body towards the axon terminals, progressively depolarizing and then repolarizing the cell as that electronic potential continues down the axon. So there is a little bit of a refractory period at the end of the repolarization. So when one neuron fires, it causes the neurons around it typically to fire as well. And then this wave of depolarization moves along the axon away from the stimulus known as a nerve impulse. Until the resting potential has been restored, a second action potential cannot be conducted. So a nerve can fire one time, has to go through its refractory period, back to its resting potential, and then it can fire again. Now, this is at least 1 millisecond long due to the fact that for this time, the membrane cannot be made permeable to sodium, so the ions cannot cause depolarization. And the stronger the impulse, the longer it takes time to recover. So a nerve can only fire so often. There has to be a refractory period while it repolarizes in between. So a nerve impulse is a chemical event. Once the impulse has traveled through the entire length of the axon, it reaches the axon terminal. And the axon terminal is going to be in close contact with the dendrites of another neuron. And the space between the axon terminal of one neuron and the dendrites of another is known as the synapse. Now, the axon terminal is also can be known as the presynaptic neuron, and the dendrites of the neuron that will be receiving the transmission can also be known as the postsynaptic neuron. So at that axon terminal, there are certain synaptic vesicles, mitochondria to provide energy, and neurotransmitters that are present, which can act as messengers to move that nerve impulse across that gap from one neuron to another. So neurotransmitters are needed to carry the message from the axon terminal to the dendrite. So that synapse is the tiny gap between the neuron and the next cell, which would be the postsynaptic neuron. And there are two types of neurotransmitters. We have excitatory neurotransmitters, such as acetylcholine, which causes the sodium channels of the postsynaptic membrane to open the enzyme cholinesterase or acetylcholinesterase. And that acetylcholinesterase is released after acetylcholine, so it breaks down so that acetylcholine doesn't continually try to stimulate the postsynaptic neuron. So acetylcholine will be released from the axon terminals. It will cross that synapse. It will stimulate the postsynaptic neuron or the dendrites. And then acetylcholinesterase will be released to break down the acetylcholine so it doesn't continually stimulate that neuron. Some of the acetylcholine is also going to be reabsorbed back into the presynaptic neuron to help it clear out of the synapse. Interestingly, insecticides will block the action of cholinesterase. So insecticides do their job by preventing the reabsorption of acetylcholine, basically causing animals to have so many different neural impulses that they can't function. And then that's how it kills the insects, which is why it's a very dangerous chemical to use. So acetylcholine in general is responsible for alertness, learning, and memory. And it's being researched for its possible role in Alzheimer's diseases. Norepinephrine or epinephrine is also an excitatory neurotransmitter. And upon its release, the postsynaptic neuron is excited. It will send a neural impulse, but then it will be also removed from the synapse through reuptake. But we also have inhibitory neurotransmitters, such as Gaba, and these have a hyperpolarizing effect. So these neurotransmitters make the postsynaptic membrane more permeable to potassium rather than sodium. So they make the postsynaptic neuron less likely to fire. So by opening more potassium gates and letting potassium out of the cell, it increases the number of potassium ions outside relative to the inside. Like I said, to make it more difficult to generate an action potential. So problems with GABA are found in people who have epilepsy, Huntington's disease, and some people with sleep disorders because their neurons are less likely to fire. So, when we look at the speed of nerve impulses, different nerves can send messages at different speeds depending on their function, location in their body, the axon diameter, if it's myelinated or unmyelinated. So, myelinated neurons have a faster transmission speed than unmyelinated neurons, and central nervous system fibers are unmyelinated, um, and they have a small diameter and they move the slowest. C nerve fibers, sorry. A delta and A beta nerve fibers are both myelinated, but A beta nerve fibers have a larger diameter, so then therefore they would be slower than A delta. When we look at a nerve impulse, we typically have an all or none response threshold. So a stimulus must be large enough to start an impulse. It must reach the threshold level or the minimum stimulus to produce a response. And every axon is going to have its own threshold level, and it's going to be unique to that axon. Increasing the stimulus strength does not increase the impulse strength. A neuron will either fire or not fire. So this is an all or none response. So once that threshold level is reached, that impulse nerve will send an impulse. If the threshold level is not reached, no nerve impulse is sent. The intensity of this stimulus instead is detected by our bodies by the increase of frequency of nerve impulses. So a warm glass rod will send nerve impulses at a slower rate compared to a high glass rod. Also, the amount of nerve. So if we have a large um, area that is being stimulated versus a small area that's being stimulated. So here we can see that um, the threshold stimuli on the bottom in the pink bar graph versus the nerve impulse on the top green. So we have that sub-threshold stimuli in the first two where we don't have a nerve impulse reached. Then we have our third one is the threshold stimulus. We can see the strength of our nerve impulse. And even if we have a super threshold stimulus, our nerve impulse is only so far. So I also want to talk about the effects of summation. So some neurons require impulses from many other neurons in order to fire. So this is called summation. So a neuron that needs summation will collect nerve impulses from many different neurons before it fires. So it will have a much higher threshold for stimulus before it will fire. So when we look at the factors influencing the speed of conduction, we can look at the diameter of the axon. The larger the diameter, the faster the rate of conduction. Myelinated axons, because the myelin stops that ion movement across it, the action potentials are only generated at the nodes of Ranvier, which is called saltatory conduction, which speeds up the rate of nerve conduction. Thank you for joining me today to talk about the nervous system. I look forward to seeing you guys next time. Bye.